Hello, can you hear me? Yes, good. Uh, oh, sorry.
Unmute.
Yes, great. Fantastic that you're here.
Yeah, thanks for having me again.
I'm going to make you a co-host.
Okay.
So now you can also share your screen and stuff. Um, so do you mind if I record the session?
Go ahead.
Go ahead. Okay, thanks. I'm going to admit everybody soon. So
Yeah,
you can already set up your presentation if you like.
yeah. How is it going with the online
Yeah,
lecture?
that's something different, right? But I think in some sense it's going quite all right. The only thing is that I had to change the exam to 9 questions after each exam. So
Yeah,
there's quite. Um, this is the same presentation as last year, right?
yeah. It's almost the same.
Yeah. So I think like we need to keep it the timing at 60 minutes. So
Yeah,
maybe you can skip some of the slides that are less essential to the key message.
yeah,
I think the core thing is to show what you've been doing recently. That would be the most fun. And when it comes down to visual prosthetics, that's I think the main or the key thing that they want to see.
Ah, okay.
So great. Yeah,
Okay. I will try. Can you see my presentation?
yeah, definitely.
Okay, great.
I'm probably going to admit everybody if you
Yeah,
don't mind.
yeah.
Okay. Hello, everybody. We're going to start in a couple of in 2 minutes or so.
Are people's videos not on?
No, that's correct. They have to turn it on themselves. So they can if they like. Hi.
Hey.
Okay. Yes, it's 1 o'clock. So can everybody hear us? Yes. Okay, great. I want to record this session. Does anybody object to this? Then I won't. Just say yes if you object. No objection. Fine. If you keep your video off, then at least you won't show up. So, okay, great. Then I'm going to start the recording. Excellent. So, Singh, welcome here to the Adaptive Brain. Thank you so much for being here as one of our guest lecturers. To everybody here, if we're talking about science fiction, this is actually what Singh is working on already. So, she's going to show you the current use of neuroprosthesis So basically how you can enable especially vision in blind people. So please take it away, Sing.
Okay, everybody, thanks a lot for being here. I'm gonna dive straight into it because there's quite a lot to cover. So I'm going to give you an overview of neuroprostheses, how to interface with the human brain. And I want to start with something fun. So we're going to have a short quiz. I'm going to show a picture on the screen, and you have to decide whether you think that this is fact or fiction. So if you think that this picture comes from a movie, for example, then okay, that's fiction. If you think that it's from real life, then it's fact. So here goes the first picture. Okay, so I don't see your videos, most of you, but if you think that this is fiction, then raise your right hand. And if you think that it's fact, raise your left hand.
I'm going to put a quiz in. So
Okay. Okay, so I hope everyone is just doing it at home. And the answer to this is that it is fiction. This is from a movie called The Matrix, which is from the 90s. And it's about a time in the future when machines have taken over the world and they're able to actually use humans to create electricity. So humans have been brainwashed. Okay, next picture.
okay, the poll is launched.
Okay, lots of people think that it's...
Almost everybody thinks it's fiction. 95%. 96% thinks it's
Is
fiction.
this for the previous picture or for this picture?
For this picture.
For this one, okay. Okay, so yeah, most people have voted. I'm gonna go ahead. This is from a movie called Ghost in the Shell from 2017. And this is also about a kind of dystopian future in which the people have basically been fused together with robots. So they're kind of these cyberkinetic beings. And they can even replace limbs if you get badly injured. Okay, so that's fiction. Let's go on to...
Yes, the poll is launched again.
Yep.
Everybody is saying that it's fact.
Yeah. And everybody is right. So this is actually from a study conducted in Utrecht by the group of Nick Ramsey, in which they actually created a first of its kind take-home clinical implant for a patient with ALS. So she had locked-in syndrome and she could only move her eyes a little bit to communicate. And so with this implant, she was able to control a wireless interface and communicate by spelling out words on a keyboard. So that is real. Okay, how about this one?
Okay, relaunching the poll. Lots of everybody is saying that it's a fact.
Yeah, well, everybody is right again. So you're doing very well. This was a study carried out at the MGH with a patient who had a motor prosthesis. And this allowed the researchers to read out his thoughts, especially his movement thoughts from the motor cortex, and control a cursor on a screen and interact with a computer. So that's very, very cool technology there. Okay, how about this one?
You can vote again. Now it's 60-40. Most people think it's fiction.
That's cool. Okay, so I would be really curious to know like why people think it's fact or fiction, but the answer is that this is real. This is from a study carried out from 2002 to 2004, the Dobell Artificial Vision Study, in which A bunch of patients with complete blindness received brain implants in the visual cortex, and they were able to see very simple shapes and forms, such as the outlines of people, houses, roads, and other objects around them. So this is back almost 15 years ago now. So that's actually real. Okay, how about this one?
Okay, there we go again. Paul. 86, 87, 88% fiction.
Yeah. Okay, well, that was fast. Yeah, you're right. This is fiction. It's from a movie from 95, and it's set in the US, in which people are able to wear these devices that look like EEG devices on their heads. And they can record their memories using these devices, and they can also do playback. So they basically wear the device and they can experience someone else's memory. So this is fiction. Okay.
Okay, everybody's voting again.
Yeah.
94, 90% fiction, 91, 92% fiction.
Okay, so that's mostly fiction. And again, that's right. That's from a movie, Surrogates, in which people actually use avatars to move around outside. And the world is a much safer place because people don't leave their house. They just lie in bed all day and control these robotic avatars that look a lot more attractive than they are in real life. Okay, how about this one?
Okay, 100% fact is coming in.
Okay, I think that's pretty clear that this is fact. So this is from the University of Pittsburgh as part of their BrainGate study, in which these patients were able to control a robotic arm with their thoughts. So this is a lot more complicated. It's not just controlling a cursor on a screen, but it's really being able to control this mechanical arm in 3 dimensions. So that's
Can I ask you something about the bilateral blindness picture? Are those people allowed to walk by themselves? Can they walk by themselves outside, or do they need like someone to guide them or a dog to guide them?
Yeah, it varied a lot from patient to patient. One of the best cases was the guy in that picture, Jens Naumann. And he actually does a lot of navigation by himself with or without the implant.
Okay.
So he uses a cane and he doesn't use a dog. So it also depends a lot on the individual.
Okay, thanks.
Yeah. Okay, I think this is the last one.
Okay, the relaunching the poll one more time. Yeah, there we go.
Facts.
Yeah.
Wow, it's really good. Everyone is acing this. Okay, so this is indeed fact. This is Jeroen Berg. He's one of the 3 people in the Netherlands with a retinal implant, which he received some years ago. And it allows him to actually identify objects and navigate more independently. He does use a blind dog, a guide dog. Okay, so this gives you an overview of what's out there, what's possible. And the reason why I start with this very sci-fi sort of poppy introduction is I think that we're all inspired, a society as a whole and also scientists, by science fiction. We're inspired by art and creative things. And I wanted you to think a little bit about the boundaries between what is fact and what is fiction. Because if you think about it, these technologies have already been developed to some extent in the lab. But then the question remains, where are all the bionic people? How come we don't have lots of people walking around? Why are there still blind people? Why are there still paralyzed people? And I think every time you read an article online, if it's not published in a scientific journal, maybe it's on Reddit or something, it's easy for the media to distort or overrepresent the results that you can get in a lab. And that can be very difficult because then it's hard for the public to understand what the limitations of the study are. What are the real advances? why is it so difficult to develop a clinical product? And so I want to get you thinking a little bit about the challenges involved in translating what you saw in the lab, for example, the people with the robotic hand, and why is it so difficult to bring this into reality and have people just walking around with a hand? So what has actually already been solved and what still remains to be solved? Okay, so I'm going to give you an overview of BCIs and talk about some real world applications as well as lab research. And generally, I've divided the applications into the different systems, auditory, motor, somatosensory, and visual. So first off, what is a neuroprosthesis? That's a device or system that makes a connection and communicates with the nervous system. And when I say nervous system, this could be the central or the peripheral nervous system. So just a quick recap, the central nervous system consists of the brain and the spinal cord. The peripheral nervous system, it's the nerves, basically mostly on the periphery. And zooming into the brain-computer interfaces, these are devices that communicate specifically with the brain. And you can ask yourself, okay, are these devices used for fundamental research in the lab? Because then probably they require a lot of collaboration. They need a team of scientists to set up. It's very complicated to get them to work. Or how about a clinical product? If they're a clinical product, then they need to be reliable, they need to be durable, they need to be working safely in humans. And how long can they work? Is that on the order of days, weeks, or years? For a clinical product, that should ideally be years. Where is the implant located? So again, I mentioned a few different cortices, sensory cortex, motor cortex, or somewhere else. And here you see an overview of the brain. I'm sure you know that there's different lobes in the brain, the frontal lobe with a lot of higher order cognition, temporal lobe, including hearing and smell, occipital lobe with a lot of visual input and processing, motor cortex, somatosensory cortex. And so if you implant a specific part of the brain, then you have a brain prosthesis that does something which affects that particular type of processing. Okay, so I'm going to introduce two very important concepts, and that is recording of brain activity versus modulation of brain activity. And so when I say recording of brain activity, that's reading out the activity that's going on in the brain during its natural state. And modulation of activity, that's also known as writing to the brain. That means that you're imposing your will upon the brain and making the neurons do something that you want them to do. And so how exactly does recording of brain activity work? Well, basically, normally signals propagate through the brain through a combination of electrical charges moving through the neurons and chemicals that send information through the synapse across neurons. And so the neurons actually generate this electric field. And when you insert an electrode next to the neurons, you can actually detect changes in this electric field and you get a voltage trace that basically looks like signals moving up and down. And by analyzing this data, the voltage trace, you can tell whether the neurons are being more or less active. So that's how you record brain activity and work out what the neurons are doing. So here you can see an image that's a neuron, that's a cell body, and there's a microelectrode that's inserted near to the cell body. And as you can see, it picks up signals from the cells around it. So from this one, and a few others. And the closer the cells are to it, the stronger their contribution will be to the signal that you record. Okay, so that was brain reading. How about brain writing? How does that work? So basically, when you stick an electrode into the brain, you can inject a small amount of electrical current and you disrupt the electric field in the vicinity of the neurons. And so this spread of current occurs in a small volume around the electrode. And then that makes the neurons activated if their processes are close enough and they are within this volume that's activated. So here, okay, this is a cell body of a neuron. And these are some of its processes. And imagine that I have a stimulating electrode S, and I put it close to the neuron, and I put it at location A, and I inject current into the brain. So that means that I'm activating this part of the neuron. If I put the electrode a little bit closer at B or C, then I'll be activating mostly this part of the neuron. And let's say that I have a second electrode, a recording electrode, so I'm not injecting current through this electrode, but I'm just reading out from the brain. I can actually measure the activity of the neuron and check whether the neuron is being active. So here's a stimulating electrode to inject current. Here's a recording electrode to understand what the neuron is doing. Okay, so here's an example, just to drive home the point from the motor cortex. Let's say we have this neuron A, and normally it's responsible for sending a command to the muscles. So for example, let's say you want to reach out and grab your cup. You think, okay, move your hand, and then this neuron sends a command to your hand muscles, and you go out and reach for your cup. Now, what actually happens during this process, imagine that I have a recording electrode next to the neuron, and I can actually read out the signal that is obtained when I'm thinking, move my hand. So I can see that this neuron actually becomes more active during that thought. Now what happens if I stimulate the neuron using this electrode, then that neuron becomes stimulated because the charge passes from the electrode to the neuron. And you can think of this as hijacking the neuron. So basically the neuron is usually used to move your hand, but now you're doing something different. You're forcing it to become active. Even though you're not thinking about moving a hand, you're just stimulating the neuron. So you're basically taking control of the neuron, and that makes your hand move involuntarily without you issuing the command through your thoughts. If someone sticks the electrode in the right place and stimulates, then your hand will just move by itself. Okay, so here's some further reading for those who are interested. And now the question is, in what applications do you use for reading and for writing? It really depends on the interactions with your external world. So for example, if you want to control a computer with your mind, is that going to be more reading or writing? If you want to move objects, pick them up, maybe manipulate a robotic hand, is that going to be more reading from the brain or writing to the brain? If you want to feel the objects, are you reading from the brain or writing to the brain? What if you want to see the objects? Or hear something? Reading or writing? Okay, so I think the answer is that you can use a combination of reading and writing for each of these applications, but predominantly, if you want to control something with your mind, or you want to read out signals, for example, from the motor cortex in order to move objects, then that's predominantly going to be reading from the brain, because you need to read out the motor commands. And if you want to be able to touch the objects, to feel them, hear them, and see them, then that's mostly going to be writing to the brain, because then you have sensors, let's say on a camera or touch detectors on a robotic hand, and you can feel the objects or see the objects if you stimulate the brain. Okay, I hope that was clear, the distinction between reading and writing and how they are used. So I'm going to go very quickly through a different modality, which is hearing. So cochlear implants, they interface with the peripheral nervous system, so not with the brain. And basically how they work is you have an implant inside the cochlea. You also have an external apparatus that picks up sound in your environment, and it translates it into a series of instructions, signals are sent to a wireless device that's implanted. And this device basically instructs the cochlear implant to send signals to your cochlea. So you're stimulating the cochlea. And that's how people who are deaf are able to hear again. Okay. Now, the thing is that these implants require an intact cochlear and auditory nerve. So what happens in people where that pathway is damaged, then it's not possible to actually stimulate the cochlea anymore because it doesn't work. So there's another device which interfaces directly with the brainstem, auditory brainstem implants. So this is a next generation device, and that is a possible solution for people who do not have functional cochlears. Okay, so just to quickly go through the difference between a cochlear implant in the peripheral nervous system versus an auditory brainstem implant in the central nervous system. The cochlear implant interfaces with the cochlear, so that's this part here. Whereas the brainstem implant interfaces with the brainstem, which is this part here. So this is in the periphery, and this is in the central nervous system. Okay. So why do we have these different types of implants? What are the advantages and disadvantages of interacting with peripheral versus the central nervous system? I think this is something that's quite interesting. And obviously, like I just mentioned, it can depend on the degree of damage. If your cochlear is too badly damaged or not usable, or you don't even have cochlears, then you can't interface with the peripheral nervous system. You have to interface with the central. Well, and then here are some other points about interfacing with the peripheral nervous system. One thing is that it's less invasive because you interface with nerves, not directly with the brain, so it's somewhat less risky. It's also easier to access surgically. It's typically less costly. And typically, it means that the technology is somewhat easier to develop and test. If you need to go to the central nervous system, you need to access the brain through the skull, and that means a risk of brain infection, which can be very serious. You need to develop and test technology that interfaces with the brain, so there's a higher barrier for testing. There are more regulatory hurdles, it's more costly. So just to give you an insight, cochlear implants have been around since the 60s, the hundreds and thousands of people walking around with them, whereas the number of people with auditory brainstem implants, that's a few thousand. But in both of these cases, the equipment is fairly well developed. It can be used at home, outside the hospital, outside the research lab. Okay, so those were applications which already exist. Now we're going to dive into research that's more experimental. So here is a technology called the Utah Array, which was developed in the lab of Richard Norman. So it consists of these very tiny silicon needles. As you can see, this is an array of 10 by 10 needles, so there's 100 of them. And each of these is just a few hundred microns in diameter, and they're about 1 millimeter long. So these needles are inserted into the cortical tissue, and this device has been approved for implantation in people. So here is an application which was developed in monkeys previously, in which they implanted the Utah array inside the motor cortex of the monkey. And they read out the signal, brain reading. They decoded the signal so that they could extract the intention of the monkey to move its hand. And the monkey was trained on this task, and it ended up being able to control a cursor on a screen without actually using the joystick. Okay, so I'm going to give you a very quick feel of how it looks in the lab.
At the University of Pittsburgh, implanting electrodes inside the brains of monkeys.
This is an array.
Andy Schwartz, a neuroscientist at the university, implanted this grid of electrodes. It's tiny, but there are 100 sensors, each listening to a different brain cell or neuron. What does it sound like?
Well, we can turn it on. It sounds like a Geiger counter clicking.
Can we turn up the speakers? It's like listening to the symphony of the brain, but now sitting in the front row. So tell me what we're hearing.
Right there, you're hearing the cell fire.
The brain is telling us how it works. We just have to figure out the language. Schwartz has been decoding that language by watching the monkey's movement and recording the corresponding signals in its brain. So what does that tell you?
So there's a relationship between how fast the neuron fires and the way the animal moves its hand. We're trying to understand that relationship so that if we see a neuron firing, we can say, the animal is about to make this kind of movement.
Once Schwartz started to figure out that relationship, he was able to connect the monkey's brain directly to a robotic arm. Within days, the monkey operated the arm as if it was his own.
The monkey has both arms restrained, and we're recording brain signals from its brain, and it's using those brain signals to operate this entire arm.
Okay, so that gives you an idea of how the research was conducted first in monkeys. And here is an article for further reading. And this resulted in 2004 in a clinical trial in humans in which Matthew Nagel was the first person to use a BCI to restore functionality due to paralysis. So this is him, you saw that picture earlier, and he was able to control the movement of a cursor using an implant that was implanted in motor cortex. So he could operate a TV and check his email, and he could even open and close a prosthetic hand. And now this is a more recent study from 2011 and 12 with these two participants. They were able to control a robotic hand to drink and even to feed themselves chocolate.
Can I ask one question? The question is, in brain reading, do they use deep learning algorithms to decode the signals?
Yeah, they do quite often do that. It depends a little bit on how much data they can get and whether it's meaningful enough to be able to run these very powerful machine learning techniques. And so in the beginning of the implantation, especially in the weeks following implantation, you tend to get very good recordings. And let's say you can get them on a lot of channels, so 100 out of 100. And then you can throw really powerful CNNs, neural networks, to decode the data. And with time, the signals tend to decline a little bit. So you can even use machine learning to try and predict the way in which the signals decline and try and exploit the amount of information that you can extract from the signals. But I do have to say that I know some folks who were doing research in Newcastle on the motor cortex, and the monkey was able to control a cursor and they recorded from only 2 neurons. So it's possible to do that kind of decoding even without a lot of powerful computation sometimes.
Thanks.
Okay, so this is the study from Utrecht University with the patient who had ALS and locked-in syndrome. And so she was at first able to communicate just using eye movements, but otherwise she was completely paralyzed. And they implanted subdural electrodes over the hand area of the motor cortex. And they also had a wireless transmitter to send the signals out to the external equipment. And so if she wanted to control the cursor on the screen, she tried to think of moving her right hand. She couldn't actually move her right hand, but she just thought of the action. And to move the cursor downward, she would try to think of just relaxing her hand. So she learned to select items in rows and columns on the screen. And she basically had to try and move her hand for approximately 1 second in order to click. So she was making brain clicks instead of mouse clicks. And you can read more about it or check out a YouTube video. Okay, so now let's move to something that's a little more complex. We were looking at motor prostheses just now, where you read signals out from the motor cortex. And what happens when you combine them with sensory feedback? And so previously we had the monkey with the implant in the motor cortex, and it could control a cursor. Now, what if we send signals about the tactile information? Let's say when you hold an object and you can feel the amount of torque or force of your grip on the object, or even the texture of the object. What if you could send that information back into the brain so that you knew whether you were actually in contact with the object and how much pressure you are exerting on it and how the object felt? And so again, to recap, first, you have an implant in motor cortex, primary motor cortex. You record signals and you can decode the signals to control a robot arm. But the thing is, it's extremely difficult to actually hold onto anything with a robot arm if you don't have the feedback from the arm. So researchers developed these sensors on the arm, and they would basically decode those signals, convert them into a kind of code that the brain could understand, and then they would send this feedback back to the somatosensory cortex as one, and stimulate somatosensory cortex at exactly the right places with the right amplitudes in order for the person to be able to tell what the arm was doing. And so in order to do this, they first started, again, in monkeys. They mapped out the somatosensory cortex, so the hand area in monkeys, and they could see, okay, in this part of the brain, certain regions code for the thumb, certain regions code for the index finger, and so on. And so they knew which parts of the brain to stimulate in order to give the monkey the sensation that there was something in contact with its fingers. Okay, so.
Can I ask one question? So how long does training generally take when you're doing these procedures?
Well, okay, so training begins with getting the monkey customized to doing tasks, very simple tasks. That can take several months. And then you actually have to train it on more specific and complicated tasks. So for example, to move a cursor on a screen, that could easily take several more months. So I would say give it at least half a year to a year.
Thanks.
Professors, scientists, and astronauts. It's the White House Frontiers Conference at Pitt. Among the guests, Nathan Copeland of Dunbar, paralyzed from the chest down in a car accident 12 years ago. Following brain surgery, Pitt and UPMC collaborated to produce a robotic arm which can be moved by his thoughts and for the first time includes the sense of touch.
We're implanting electrodes, these little grids of electrodes into the brain, and using these devices, this person is able to reach out with a robotic arm, control the movement, grab objects. And not only that, we can generate sensations that feel like they come from his own hand.
He volunteered generations.
There's already not a lot of people that could do it. So when they asked me, it was basically like, why not? Like, why would I not do it? So eventually this will help tons of people. For now, I get memories and I get to do cool stuff.
A crowd gathers on Fifth Avenue. A special guest is coming. What could better exemplify the importance of that technology, plus other exhibits here at Alumni Hall, than a visit from the President of the United States? Led by a phalanx of motorcycles, the presidential limo makes the turn toward Alumni Hall. Nathan Copeland, meet the president.
And the sensation you receive when we are shaking hands.
Yeah, anytime there's torque on the hand right now, I will receive sensation.
Barack Obama's shaken many hands, but with a fist bump to remember. I'm Dave Crawley.
Okay, so that was the big highlight, the study in 2016, where they were able not only to read out from motor cortex, but also write to somatosensory cortex in humans. So now let's move on to spinal cord prostheses, which are combined with motor prostheses. So spinal cord injury affects about 50,000 people each year. And there's damage to your spinal cord, which means that even though your brain is generating instructions, and you want to move your limbs, the signals do not get down to your limbs because they're disrupted at the level of the spinal cord. So here's a prosthesis that was developed in Switzerland in rats first. So they received a lesion of the spinal cord, which basically disrupted the transmission of signals. The researchers injected a pharmacological mixture into the spinal cord in order to stimulate firing of the neurons. And importantly, they implanted stimulating electrodes into the spinal cord that would bypass the site of injury and send the brain signals directly to the part of the spinal cord below the injured area. So this was known as an electrochemical neuroprosthesis. And here you can see approximately how it works. The mouse is supported on a treadmill, and the researchers measured its gait, so its stepping motions. They had this electrochemical neuroprosthesis where they injected serotonin, and they also had these electrodes implanted to be able to stimulate the spinal cord. And here I'm going to play this, just part of it to let you see how it looks.
With this in mind, we developed a completely new robotic system to support the rat in any direction of space. Imagine, this is really cool. So imagine the little 200-gram rat attached at the extremity of this 200-kilo robot, but the rat does not feel the robot. The robot is transparent, just like you would hold a young child during the first insecure steps. Let me summarize. The rat received a paralyzing lesion of the spinal cord. The electrochemical neuroprostheses enable highly functional state of the spinal locomotor networks. The robot provides a safe environment to allow the rat to attempt anything to engage the paralyzed legs. And for motivation, we use what I think is the more powerful pharmacology of Switzerland. Fine Swiss chocolate. Actually, the first results were very, very, very disappointing. Here is my best physical therapist. Completely failing to encourage the rat to take a single step, whereas the same rat 5 minutes earlier walked beautifully on the treadmill. We were so frustrated. But you know, one of the most essential qualities of a scientist is perseverance. We insisted, we refined our paradigm, and after several months of training, the otherwise paralyzed rat could stand. And whenever she decided, initiated full weight-bearing locomotion to sprint toward the reward.
Okay, so I am going to, I think, very quickly show the follow-up to the experiment in rats, where they did something very similar in monkeys.
We restored locomotion in primates with spinal cord injury. And for this, we developed a brain-spinal interface that decodes motor intention from brain signal, bypasses the lesion, sending this information to the spinal cord below the injury in order to reproduce the intended walking movement.
For the first time, I can imagine a patient completely pelagic being able to obey to his brain commands through this brain-spinal interface. If you have a lesion of the spinal cord, then the command is not going to reach the legs and going to stop at the level of the spinal cord lesion, and the primate is not going to walk.
So concretely, we inserted 100 electrodes in a small region of the cortex that control the leg and send the information from all the neurons we recorded to a computer that decoded the motor intention of the primates based on this signal. This means the extension or flexion movement of the leg. And the computer sends then this information to an implantable stimulator that had the capacity to deliver stimulation at the correct location, the correct timing, in order to reproduce the intended extension and flexion movement of the leg.
Okay, so I'm not showing the whole thing. You can always watch it later. But I wanted to give you an idea of the progression in these studies in the lab, how they go from the concept to doing the test in rodents, and then if that works, trying it in monkeys, and then hopefully making it into patients. And their group has now a clinical trial going on with patients with spinal cord injury. And so this kind of gives you a little behind the scenes taste into how you actually develop something for the clinic. Okay, so now my favorite topic, let's move on to visual prostheses. I'm going to start with retinal prostheses. And here is an overview of how the visual system works. The light comes in through your eye, it hits the retina, where these light signals are converted into electrical signals. They are sent through the lateral geniculate nucleus, that's this area in the center of the brain. And then they're sent on further to the primary visual cortex, which is in the back of the occipital lobe. So that in a nutshell is how the visual signals are sent. And you can imagine that if you want to interface with the visual system, then you could do that at one of several different areas. This is an image from one of the articles that you had as your assigned reading. You can have a retinal prosthesis, you can have an optic nerve prosthesis, one in the LGN, or one somewhere in the visual cortex. And there's pros and cons to each of these approaches, but people are working on every single one of them. So a lot of blindness is caused by retinal disease, for example, macular degeneration or retinopathy. And remember that the retina is peripheral, so it's not part of the central nervous system. And this means that it's a little less invasive, and retinal-based therapies are quite a bit more advanced than cortex-based therapies at the moment. Here is how it works. So imagine that you have a blind patient. They're wearing a camera on a pair of glasses. The camera records everything in your visual field. It sends these signals to a pocket processor, which is outside this picture. The pocket processor uses different algorithms, could be machine learning, for example, to generate basically more simplified objects out of these pictures. It sends these into a wireless transmitter, which is implanted. And these signals are then delivered to an implant, which is on the retina, which stimulates the cells that remain on the retina, and then allows people to be able to perceive the forms of objects and detect light versus dark. And so here again, this is Jeroen Perk. He's given a lot of interviews and there's a lot of articles from him that talk about his experiences with the device. This is how the device looks when it's implanted in the retina. There's 60 electrodes, which in principle can lead to 60 pixels. And okay, I think we have time to watch a little bit of this.
It was emotional. I was laughing because I was very happy with the light and with the skiing, and yeah, that's the most important thing. And I felt a little bit crying because I achieved my goals with the system. So that makes me a happy man. Yeah. I am Jeroen Perk. I'm coming from the Netherlands, from Amsterdam. I got the diagnosis on my 11th. I got retinitis pigmentosa, and at 18, 19, I lost 80-90% of my visual field. Today I'm here because I was skiing, and I do it with the Arcus 2 system. That's the glasses with the camera on it and a chip in my eye. The Arcus 2 system. I think it's a new chapter in my life. I got some visual fields back, and yeah, nowadays we did the archery shootings and we did the skiing, and yeah, I think.
So,
one question is, how much delay is there with a prosthesis like this?
I think it's very small because it stimulates with a certain frequency. I'm not sure exactly, maybe 3 times a second or more. And so it's enough for them to be able to track movement and be able to move around as well. And the one thing is that since there is this stimulation frequency, they don't see a continuous picture, but it's almost like strobes. So they see this thing flickering at maybe, you know, whatever frequency, 3 times a second. And they say that in the beginning, after surgery, it can be very distracting to have this thing kind of pulsating away, but after a while, they get used to it. Okay, so the nice thing about this video was, did you notice that he was following a guide? And the thing is that this system, it's quite simple. It's only 60 pixels, but it's good for light detection. And so the thing is, he was skiing on the snow, and the snow is very light in color, and the guide was high contrast against the snow. So it was very easy for him to see this guide. And these conditions are actually optimal for the working of this kind of prosthesis. Yeah, it only has 60 electrodes, not 16. It gives some light and dark perception. You wear a camera. It's powered by a battery. And so all of these, all of these technologies for the retina are a little bit limited in the sense that if you have a person whose retina is completely non-functional, or if they don't have eyes, then it's not going to be possible for them to wear a retinal prosthesis. So that's why you need to go to the cortex, directly to the brain. And so here is an overview of how the visual system works. You have your image that you're looking at in a normal person, a sighted person. So imagine that you're looking at a tree and a bunch of house. Then this image is actually processed by the visual cortex at the back of your brain. And there's a map in your visual cortex, which is flipped from top to bottom and left to right. So there's a bunch of cells over here, and the ones on the left side, they are processing the image, which is on your right side. So the stuff on the right gets processed on the left, and the house gets processed on the right side of the brain. So it's important to remember that this is retinotopic map in the visual cortex. So imagine that you're looking at this kind of stimulus and you're looking straight in the center. So you have this light blue color in the center and these concentric colored rings. And so this is an image from an actual MRI study where this is the center, that's this spot. And the neurons in the back of the brain, they process the very center. And then as you go further out, more anterior in the brain, these neurons are processing parts of the stimulus that are more peripheral in the visual field. So as you go from the center outwards, the processing of the signals goes from the most posterior part of the brain to areas that are more anterior. So now if you insert an electrode into the back of the brain and you stimulate electrically, then you'll see an artificial dot of light, which is called a phosphene, at the center of your visual field. If you move your electrodes a little bit more anterior, then this phosphene will appear at a location that is more peripheral in your visual field. And if you have an implant that has a ton of electrodes and you stimulate on all of them, then in principle, you could create a lot of phosphenes. And if you select intelligently a group of electrodes on which to stimulate, then in principle, you could create forms such as a line. So this technology has been around since the 60s and 70s with massive implants. It's been refined throughout the years and more studies have been carried out in 2000s. And most recently, there's been a study from the Orion, the makers of the retinal implants. And there's also been another study in Spain using the Utah array electrodes. And importantly, the study in the US, the Orion, that one is a clinical trial. So they had, I think, 6 to 7 patients enrolled. And these patients were actually able to recognize a very simple visual process One of them has written a book about his experiences, and 2 of them keep blogs. And it's really interesting to read that, for example, there was one patient who was able to sort his laundry, light versus dark colors, using this brain implant. The study in Spain from the lab of Eduardo Fernandez, so far there has been 1 patient enrolled in it. And this is actually a clinical research study, so it's not meant to create a real product for blind people, but it's more to understand the nature of the electrical stimulation and to bring about more scientific progress in this area. And so this is not solely for the benefit of patients, but it's focused more on furthering the field of research. Okay, I'm going to skip those videos, but this gives you an idea of what Jens Naumann was able to see. So he is a patient from 2002, whom you saw earlier during the pop quiz. And so he was actually able to tell the difference between a cup and a bowl on a table. And this is how it works. You basically have a video feed that is converted into very simple outlines, and instructions are sent to the brain. And this is a rendering of how the stimuli would end up looking if you're a blind person using this kind of implant. Okay, so in the last 8 minutes, let's go over the work that is done in our lab at the Netherlands Institute for Neuroscience, where we basically have monkeys who are trained to report the nature of visual percepts. And they also have a brain implant consisting of 1,000 electrodes, and they report the nature of the artificial percepts. And so this is the overview. It's very similar. You have a camera on a pair of glasses that records video footage. Instructions are sent to an implant in the brain, which sends electrical signals to the visual cortex. We use the Utah array implanted in visual cortex, and we implanted many arrays across primary visual cortex, as you can see, in 1 hemisphere in each of 2 animals. So this is how the monkey brain looks. It's a little different from the human brain, but there's a large area that is nicely on the surface and readily accessible during surgery. This is how our implant looks. It's a customized 3D printed titanium implant that I designed myself to make sure that it fits perfectly against the skull of an individual animal. And here you can see there are these gold wires that connect to the electrodes. These were actually arrays consisting of 8 by 8 electrodes, so 64 electrodes on each of 16 arrays to yield a total of more than 1,000 electrodes in the brain. And so if you stimulate here, just to recap, in this particular location of the brain, then you expect to get the phosphene at that location of the visual field. If I move my electrode along, then the location of the phosphene is expected to move accordingly. So we carried out a mock surgery to refine our surgical technique with a 3D printed skull and an agar brain. And we implanted these fake electrodes into this brain. And now here you can see the actual locations of the electrodes in each of our 2 animals. And so basically after the implantation, the only part that you see sticking out from the skin of the monkey is this interface, this pedestal. We connect the pedestal on a daily basis in the lab to our external recording equipment. And this is my lab. That's how it looks with all these computers, devices for recording neuronal signals and for stimulating the brain, and also for processing a whole lot of data. So remember this implant, you can record from the brain and you can also stimulate to the brain. So I'm going to show you how it looks when we record the brain signals first. So my monkey was trained to fixate on a red dot at the center of the screen, and I would sweep a white bar across the screen. And as you can see, as the bar moves across, there's this wave of activity that sweeps across the visual cortex. And you see that as a wave of yellow going across the cortex. That was in the first monkey, and now this is in the second monkey. It's the same task, fixate. I sweep the bar, and you see that yellow wave sweeping across the visual cortex. And as I sweep the bar in different directions, you can see that the wave of activity also passes in different directions. Okay, I carried out something which is called receptive field mapping. So I was able to analyze my data and identify the location at which each of these electrodes were in the brain, and also the location in visual space that I expected a phosphine to appear at if I stimulated on that particular electrode. And so, as you can see, I've color-coded each of these arrays of 64 electrodes, and these dots show you in visual space coordinates where those phosphines would end up being. And you can see that the locations are grouped, nicely clustered according to color, indicating that if I stimulate, for example, on array 15, then I would expect to be able to obtain phosphine at this location in the visual field. So I could select electrodes in the visual cortex and stimulate them in order to create very simple shapes and forms using artificial vision. The same is true in the second monkey. You can see the nice clustering of the locations, the expected locations of the phosphines. And so previously I showed you that movie, which was brain reading. Now let's take a look at some of the results from brain writing. So before the surgery, the monkey was trained to fixate at the center of the visual field. And I basically showed a dot somewhere on the screen, and he had to report that he saw the dot by making an eye movement to it. I was measuring his eye movements with a camera at all times, so I knew where he was looking. And on half of the trials, I didn't show a dot anywhere, and he had to just continue maintaining fixation at the center. So I knew whether he really saw a dot or not. After the surgery, instead of showing a real circle somewhere on the screen, I just left the screen blank, but I sent electrical stimulation to one of the electrodes. And my monkeys made an eye movement to a location in the visual field as though it was a real visual percept. Here is a movie to show you how it looks. The red dot shows you the center of the screen. The blue dot shows you the location of gaze of the monkey. The white dot shows you the expected location of the visual, of the phosphine percept, but in reality, there was no white dot on the screen. That's just the predicted location that I calculated based on previous measurements. And so you can see on each trial, he makes an eye movement to a location that corresponds pretty closely to the expected location of the phosphine. And when I analyzed the eye movements, I saw that they were nicely clustered as expected according to the locations that we predicted previously. And that is true in both of our animals. Then I did a more complex task in which the animal was trained on a visual task. He was able to recognize a small alphabet of letters, for example, T versus L. If I showed a T, he would have to make an eye movement to a target T. If I showed an L, he would have to make an eye movement to a target L. And after he had this implant, instead of showing a real visual stimulus of a letter on the screen, I stimulated on 10 to 15 electrodes simultaneously in order to generate what I hoped would be the percept of letters. So again, the white dots show you the locations, the predicted locations of the phosphine percepts, but they did not appear on the screen in reality. They just give you an idea of how the phosphine percept should have looked. So I stimulated a bunch of electrodes to generate an A or an L, and he reported which letter he saw. Okay, so it's time, and I'm exactly at the end of my talks. That was nicely that worked out nicely. Thank you very much for your attention. And I hope you have fun with the questions, the exam questions. Thanks to all the people in my lab and to our funders and to Ilya for having me.
Yeah, great to have you here, Xing. If there's any urgent questions left, not everybody at the same time. I had already quite some questions that I could answer based on the articles and the literature. So there's one question: are there any noteworthy non-invasive BCIs, for instance, EEG, for instance, for motor tasks?
I have seen some interesting applications, primarily at events, competitions. For example, there's something called the Cybathlon, which is held in Zurich every few years. And there the participants have EEG, and they are paralyzed to a certain extent, and they need to do virtual racing. And so they control race cars in a virtual reality software. And the thing about these types of implants is that it can be quite difficult to isolate the brain signals. You have to set up the EEG implants very specifically in order to really read brain signals. If you just buy, if one of us who is not paralyzed just buys an EEG reader, like one of these very cheap commercial ones, and puts it on our head, the thing is that these also pick up muscle artifacts. So if I'm trying to do a virtual reality race on a screen, I could be looking somewhere and using my eye movements. And instead of using my brain signals, I could just be using my motor movements. And so it's very difficult to control that you're really only using brain signals, which is why in these competitions, there are a lot of rules and checks and balances to try and isolate the brain signals and make sure that the participants are really only using their mind, their thoughts to control the signals, and not just using the eye movements or the eyebrows or something.
Thanks. One of the other question is, are they also able to identify letters which they haven't seen before and without training?
I think in principle, if you did that in human subjects, it is possible. In fact, the lab in Spain, Eduardo Fernandez's lab, they have done a study doing exactly this. So if you Google them, there's an article in the MIT Technology Review where the patient was actually able to recognize a big letter O versus a small letter O. However, this is very difficult to test in monkeys because you can't actually just verbally ask them what they're seeing. Having them report the shape of a letter requires a lot of training on visual versions of the letter. And so this is something that would be best be explored in human subjects.
Final question, is there research ongoing on visual convolutional networks to integrate some higher visual processing, for example, colors?
Yeah, I think there's a whole variety of this type of research. And so I know a few labs that are working on not just colors, but also temporal integration, looking at how signals are actually processed in the retina and then sent to the brain via the LGN. There's people working in Nijmegen at Radboud University on different models in order to sort of mimic how the brain processes visual images. There's people also working in the lab of James DiCarlo in MIT, and they also look a little bit at higher order visual areas, such as, I think, different parts of temporal cortex where the brain is more involved in object recognition. And so they do apply basically convolutional neural networks to decode the signals throughout different parts of the visual system and relate it back to how the brain works.
Okay, great. That's it for now. Well, thank you so much. Everybody is going to answer some questions based on the articles that you shared. Thank you so much for your time. It was a great pleasure to have you. And I think for these students that have to decide on their masters soon or have decided on their masters, this is quite exciting stuff that's going on in Amsterdam at the Netherlands Institute for Neuroscience. So think of this in the future. So please sing thanks, and great to have you again.
You're welcome. Good luck.
Yeah, thank you very much.
Yeah, good luck with your studies and your future and your careers.
Which master should I follow to be able to work in your lab in the future?
I think there's people who have there's different backgrounds that would be applicable. That includes neuroscience, neuropsychology, also computer science. We also take master's students for internships quite regularly. And for the most part, that is work with rodents, because we don't just work with monkeys. We also have worked with humans, so monkeys, humans, rodents. And we also have some computational neuroscientists who do machine learning. And so check out our website, get an idea of the type of work in the lab. There is definitely room for interns, and we're going to be hiring PhD students and postdocs in the future.
Nice, thanks.
Great, thank you so much. I'm going to close off. You can reach me by email or phone calls in case of emergency, and you have time until 3:10 to submit your exam question. Good luck.
Bye.
Bye.